The Center for Genomic Medicine was founded in 2010. The idea was to take advantage of new global technologies to characterize the individual patient's genomes. Our department analyzed samples from a variety of patients, mainly from cancer patients, and where we examine for hereditary dispositions, but also sporadic cancer, aiming at defining a proper drug that the patient can be treated with. There has been two changes. First of all, we can actually categorize and diagnose cancer more accurately. On the other hand, we also see a development where we can sequence the genome and define particular activating mutations that can be used for targeted treatment.
Sequencing has really moved the field forward. It's more accepted that you should look more globally for the disease to find out what's causing, what's driving the disease. I'm working with a project where we're making genomic profiles. We get samples from cancer patients where there are no other treatment options. And the point of all this is to find new treatment targets.
Our experience with the gene reader system is very good. We are focusing on some of the short turnaround times that we have on patient samples, and using this system we can have come from a sample to an answer within 5 days.
Yeah, one of the big issues by employing next generation sequencing into the clinical environment is the ability to treat the big data coming up from the analysis.
The bioinformatics is extremely important. It's usually a bottleneck in the data analysis.
We started with a pipeline with the CLC software and the Ingenuity platform. This has now been combined into one software, and that is very convenient.
So it's a very smooth system.
We are very happy to use the QIAGEN products because they are developed for clinical use. They are reproducible, easy and fast.
I think within the next couple of years we are focusing on small gene panels with short turnaround times, which is very important. But on the long run you'll probably go into whole genome sequencing.
Whole genome sequencing, there's obstacles, you can say. It takes more time.
You have all the raw data, you need to map them and call the variants, and you need to interpretate.
It's also important to consider what you're actually adding by maybe multiplying your data with hundreds. So if you're not really adding anything, we're talking about targeted treatment.
What I like is especially when you can help the oncologist to choose the right drug for the patient, and that we hear that the patient receives the drug and gets better from our work.